which has actually, done so much of testing. The topic is very relevant today as the government doing the, but not only the COVID-19 testing, but also the laboratories play a very important role in the entire healthcare system. We are pleased to invite today, and Dr. Jain agreed kindly to speak on this topic. Most of you are aware about Dr. Neeraj Jain. He is the MBBS and MD pathology from the University College of Medical Sciences in Delhi. He is also the secretary of the Association of Practicing Pathologists, apart from his other lot of accolades, and he is also the NABL and NABH assessor. So Dr. Jain, I won't take a lot of time on this. I would like to request you to kindly start your session. Dr. Jain, just to inform you, Dr. S.K. Sood actually requested that he will be, coming and listening to this. And so very shortly, I think Dr. S.K. Sood will be listening to this interesting webinar as well. Right, sir. Over to you, sir. Thank you.
Okay, first of all, I would like to thank Mr. Kamal and Cliniminds for inviting me for this talk. And this is, in fact, an opportunity for me. As Mr. Kamal has just announced, Professor Sood will also join us. Many of you may be knowing Professor Sood, and many of younger colleagues may not be knowing. He is the person who actually started or who actually promoted hematology in India. And he is one of the few persons at present in the world who has actually at some place worked with Maxwell and Winthrop also. Okay, so now without any further delay, I'm starting my presentation. This is what is the impact of COVID-19 on clinical laboratories and what is the role of laboratory accreditation. But before that, as I always do, first I would like to know whether all of you are able to hear me clearly or not. Dr. Mamita Saha, please unmute yourself.
Yes, sir.
Can you hear me?
Yes, sir.
Right. Okay. You are from Bengal.
Yes, sir.
Right. Okay. And Dr. Elango, please unmute yourself. Elango, Dr. Elango. Yes, Elango. Can you hear
Yes, sir.
me?
Yes, sir. I can hear you, sir.
Yes. Okay. Great. So now, both of you, please mute yourself. Both of you, please mute yourself. And now I'm starting our presentation. All of you, please keep yourself muted so that there will not be any extra noise, and all of you will be able to see, will be able to hear more clearly, right? So actually this discussion is not related to COVID-19. We will not discuss how we can set up our COVID-19 lab. We will discuss what is the impact of COVID-19, this present scenario of COVID-19 on our laboratory. All of you will agree with me. With this COVID-19, everybody has realized the importance of accreditation in medical laboratory. A lot of innovations are going on in our medicine. Different procedures people are developing, whether it is cardiology, whether it is gastroenterology, neurology, and so much therapeutic advancements in oncology. There is immunotherapy, target therapy. For this target therapy, there is significant role of our laboratory. Gene therapy, different organ transplantations are going on. So therefore, we also have a branch, transplant pathology, and robotics are also involved in the therapeutics. And similarly, in our laboratory also, a lot of new tests are coming, a lot of new methods are being developed. People are talking about how we can reduce our turnaround time so that reports are available within very small time frame to take any decision, point of care testing, At present in India, we are growing organism in cultures to see what is the cause of septicemia. And then we are performing this antibiotic sensitivity, what are the drugs which will be effective on a particular bacteria. But in the Europe and in USA also, people are moving from this culture growing technique to identification through molecular detection. For this, they have developed point of care testing. They are able to identify organism within 3-4 hours and what antibiotics will be effective. So all these things are developing, and very soon you may find point of care testing for this COVID-19 also. And all of us have realized in COVID-19 importance of molecular diagnostics in our laboratory. And government is also promoting, they are also telling different laboratories, you come forward and start this COVID-19 molecular testing, whether it is PCR or antigen detection through some other method, or at least do serological test. What is the relevance of a particular test? That is a separate issue. But now people are coming forward. Pre-analytical automation, samples are transported through these pneumatic chutes, etc. And in the present scenario, we have also realized when government is talking, so many other people are talking, we have to take accreditation, the importance of accreditation for our laboratories. But there are certain challenges. What are these? With this new and evolving techniques and high expectation of accuracy, so much pressure on the laboratory, you are saying this person is COVID-19 positive and sample is being sent to some other laboratory, they are saying negative, who is correct, who is not correct, reverse, sometimes reverse is the issue, or what is the sensitivity of a particular test, what is the specificity of a particular test. We will also see what is the meaning of all these terms, what is diagnostic sensitivity, what is diagnostic specificity at a later stage during this presentation. Then for certain things, quality control material is available. For certain things, this quality control material is not available or not easily available. And what is the concept? What is this EQUAS? What is this IQC? How to do all these things? How to interpret all these things? If suppose there is some outlier, how to take action? Sometimes we get confused with all these things. And compliance with regulatory requirement. Previously, we were running our laboratory, but now in this COVID-19, we have realized sometimes there are certain guidelines for Ministry of Health and Family Welfare or ICMR. You have to follow these guidelines. After 4 days, some other guidelines. Now you have to follow these guidelines. So this is also a challenge. We have to train our people. Otherwise, we were working for so many years in a set pattern. We have to train our people how to carry out all these activities, how to communicate results, how to inform government through the portal, what is the capability of a test system, that also we have to see. So much pressure on the government. Previously, they were saying we are validating this test. What is the meaning of this validation? That also we will understand. Cost is also a limiting factor. If it is very costly, it may not be acceptable to community. Interpretation of a particular test, we are saying this IgG antibody is positive or IgM antibody is positive or RT-PCR is positive. What is the meaning? I'm giving you certain examples, but this is true for everything. If suppose we are saying for HPLC something, band in the region of hemoglobin S is 24%, or band in the region of hemoglobin A2 is 34%. What is the interpretation? That is also very important. As a laboratory person, it is our responsibility to help people in the interpretation. Then pre-analytical, quality of our test is only as good as the quality of sample received. And 80 to 85% errors occur during this pre-analytical phase. It is very important to have a good control, what type of sample is required, how this sample shall be collected, how the sample shall be transported to the laboratory. And sometimes what happens, incorrect results may go undetected, and when they are reaching to patient, and then what will happen? Loss of reputation for a laboratory, and sometimes some litigation may also have to face. Now, if we see what is the role of our laboratory in clinical medicine, 70% clinical decisions are based on our reports. And our intention to release error-free report, which is meaningful for clinician, they can make a correct diagnosis, they can start correct treatment, and ultimately it is resulting in better patient care. For this, what is required? Quality assurance. Now we will understand what are these terms. We use these terms very frequently. Assurance means we are telling somebody else these requirements will be fulfilled. You will get good report from our laboratory. We are taking care of all quality related issues. This is quality assurance. Now, to fulfill this quality assurance, what is required? We should have a good quality control system. Only then we can tell somebody else, somebody can say, we can assure somebody else that we are following quality, so we should have a good quality control system. For that, we have to define our quality management system. We have to document our quality management system based on some international criteria. Then we are implementing as per our documentation, and wherever we are thinking we can further improve, we are improving and documenting and then implementing in that improved system. So that means we have to develop a well-documented and well-implemented quality management system. And sometimes what happens, we think we have very good equipments. These equipments are available only in our lab, in the whole country or the whole city. Therefore, our laboratory is very good. Or we have started laboratory in a very posh area. We have very good building, very good furniture, well-mannered, well-behaved staff. All these things are okay, appreciable. But in addition to our examination, this pre and post examination processes are also very important. If these things are not properly taken care of, what all we get is automated mess. 80 to 85% errors occur during pre-examination. Now, sometimes people say certification, sometimes people say accreditation. What is the difference? In certification, only documentation is checked. Okay, this person is MD pathology and this person is reporting histopathology here. Okay, so this is fulfilling the requirement of documentation and certification. But in accreditation, in addition to documentation, technical competence is also assessed. We see whether a particular person is able to carry out his or her duty in a required manner or not. Whether that person is some authorized signatory, some doctor, some technical staff, or non-technical staff. So in accreditation, technical competence is also assessed. And for your information, wherever you see ISO 9001, that document is for certification. And ISO 15189, it is a document for accreditation. And in our laboratory, it is better if you go for accreditation, because in this technical competence is also assessed. Ultimately, we are dealing with the life of the people. So there shall not be any compromise. Now, what is the need of this accreditation? At present, if you want to start this COVID-19, it is a regulatory requirement. It is a regulatory requirement because Ministry of Health and Family Welfare asks Indian Council of Medical Research, you ensure quality of results and tests to be performed. So they then asked many people and they thought this ISO 15189 is a document which is for accreditation in medical laboratory. And NABL is an agency which is providing this accreditation based on ISO 15189. It is an international document being followed all across the globe, whether it is College of American Pathologists in USA, or it is NATA in Australia, or UCAS in England, for that matter, any other accreditation body. So they thought, we will ask people to go for this ISO 15189 accreditation. Only then that lab will be allowed to perform this COVID-19 testing. Because this is a way of competence demonstration, and everybody will recognize, okay, that laboratory is performing this test, so this laboratory has recognition. And because ICMR is saying only these laboratories will be allowed, it is mandatory also. And when you go for accreditation, you take care of so many things. So this is stimulus for improvement also. Okay, although we are performing this particular activity in this manner, there is no non-conformance, but we can further improve our activity. This is a stimulus for improvement also. If suppose it is for your information, although many of you already knows about all these things, if you want to start COVID-19 RT-PCR, for private laboratories, accreditation is mandatory. You shall have RT-PCR for viral detection in scope. Now it is at present online assessment through video conferencing, and it is a fast process. They are providing accreditation very fast. First you have to get this accreditation, then you have to get approval from Indian Council of Medical Research, ICMR. When you are following this ISO 15189, when you are going for accreditation and you are following the requirement of accreditation, which is as per ISO 15189, there are reduction of errors in the pre-analytical processes. Your reports are more accurate because you are following processes very carefully, and you are also looking at regularly improvement in your laboratory. When you are generating a correct lab report, it is ultimately resulting in better patient care. So with this accreditation, there is standardization of processes, demonstrability of results. Your people are better managed. Whatever supply you are getting in your laboratory, that is also of better quality because you are evaluating your supplier also. And you are able to communicate with your partners, your clinical partners, in a better way. They will also understand your laboratory is accredited, you're following certain quality requirements, but there are certain limiting factors. A lot of investment is there. You have to educate your people about quality management system. You have to train them as per ISO 15189 and for many other things. You have to get your equipment calibrated. You have to purchase control for IQC or IQAS. At certain places, you have to do validation also. Then also you have to bear expenses of accreditation. And this is a time-consuming process. You have to learn all these things. Then you have to implement all these things. So these are the challenges which laboratory face. But important thing is, with this, we work with totally different mindset at a different level. Right report is issued to the right patient based on right method. We are following turnaround time also. So all these things ultimately resulting in better reputation of the lab. But for this, we have to understand ISO 15189. We have to develop understanding of this ISO 15189. For this COVID-19, we have to develop understanding of molecular diagnostics and also infection management. So now Here we will restrict our discussion to quality management system in clinical lab. How we, because this is a very big topic. You see, all these things are part of our day-to-day activity. A patient is coming to our lab with a test request. First, we have to take care of how we are collecting sample. Suppose some advisory services is required. Suppose we have to tell that patient, you have to come fasting or you have to come at a particular time. So we have to follow that. If suppose we are not performing a test in our laboratory, we are sending to some other laboratory, we have to tell our patient. So this is related to our pre-examination, and all of you will agree. All these things are important part of our laboratory. Then when we are performing test by the already validated method. We know this method is good for this particular activity. And also, we are taking adequate care of IQC and EQUAS. So we are sure whatever result we are generating in our report. Then whatever sample we send to referral laboratory, now we are receiving report. So we have to see we are receiving report within reasonable time frame, and whatever result we are communicating to patient, so we are taking adequate care of that. If suppose some advisory service is required, some interpretation of a particular test, something like that, we are telling patient how we are reporting results, what all information we are including in the report, how we are releasing result. We are following all these things. Whether our laboratory is accredited or not, we are taking care of all these things. To ensure all these things, if suppose somebody is complaining, we are taking adequate care of that. If suppose there is some non-conformance, something which should not happen but has happened, we are taking adequate care of that through corrective action, through preventive action, and our focus is on continual improvement, how we can further improve our services. For this, we do evaluation. We check certain activities, important activities, otherwise which may result in some wrong reporting. We conduct audit as per some checklist. Evaluation we see without checklist, in audit with checklist based on some criteria. And for this, we have to take care of our organization management responsibility. And time to time, management is also discussing with the laboratory people how they are managing their laboratory, and what is their quality management system, what all specialties they have included in this quality management system, how they are creating various documents, how they are maintaining. Suppose some records need to be maintained for 10 years, we are maintaining for 10 years, and we are maintaining in such a manner they are easily retrievable and retrievable in a legible manner. For this, what is required? A good care of resources, whether it is personnel or your space, well-maintained space in terms of adequacy, temperature, humidity, cleanliness, everything, well-maintained equipments, reagents and consumables, and also you are taking care of your vendors, you are evaluating them, and also you are maintaining laboratory information system in such a way to taking care of confidentiality of patient information and storing data in such a manner that is not destroyed. This is our day-to-day activity in the laboratory. First, we are receiving requests, then collecting sample, transporting sample received in the laboratory, then the sample is going to different sections of laboratory, they are registering in different sections through their laboratory information system, then the test is being performed, then results are interpreted, results are reported, critical results are immediately informed to clinicians, and then the clinician is taking action on that. For this process, to maintain this process is flow of activity. To maintain this process, what is required? We require proper procedure at each step. See, here we have defined the process. Now, how we will take care of this process? We have to define the procedure at each step, how we will take care of that, properly documented and implemented in the documented fashion. Wherever you think some improvement is required, you are improving in that area. For that, what is required? People who are working in the laboratory in any capacity, they should have proper qualification. Qualification means education background and any training or experience required. Suppose you want to start this COVID-19. For that, there is no separate educational degree. You are either MD microbiology or MD pathology. But you shall have training or experience related to this PCR. If you want to start flow cytometry, no separate education qualification, MD pathology. If you want to start for leukemia, then training or experience related to flow cytometry in leukemia. If you want to start for immunodeficiency disease, experience related to that. If you want to start for hemolytic anemia like hereditary spherocytosis or PNH, training or experience related to that. Regular training in your laboratory on various aspects, training related to biomedical waste management, training related to your actual work processes, training related to understanding of this quality management system, training related to ethics of your laboratory, training related to confidentiality of patient information. All these things are part of our day-to-day activity. Then vaccination against hepatitis B. If you can get anti-HBs titer done, that is a good laboratory practice. Check whether people who are working in your laboratory are competent to carry out an activity or not. What is competence? Ability to use acquired knowledge and skill in practical situation. Regularly conduct CME, seminars, some other training programs so that your people are able to upgrade their knowledge and skill, and there is harmonization. Through this training program, everybody will upgrade their knowledge and they will work in the similar manner. Provide adequate and appropriate personal protective equipments to all your laboratory people. All of you are following this. Whatever specimen you are collecting in a laboratory, you shall have properly filled test request form. Samples are collected in the correct container. Then samples are labeled properly. And if I ask you whether sample shall be first collected and then labeled, or it should be first labeled and then collected, many of you may say it should be first labeled and then collected, but it is international guidelines. You first collect and then label in front of patient. This is as per CLSI document. Then samples are properly mixed, then transported to the laboratory, keeping in mind within reasonable time frame at appropriate temperature, and you are also taking care of safety of the person who is carrying those samples as well as integrity of the sample. Then samples are received in the laboratory. Whatever tests are required, you have adequate and appropriate samples for those tests. Then how you are receiving reagents in the laboratory, how you are storing. You should have properly documented guidelines, and how you are carrying out lab acceptance testing. This kit is suitable for the test to be performed. Now we have to understand these terms: diagnostic sensitivity, diagnostic specificity, positive predictive value, negative predictive value. Diagnostic sensitivity means please all of you are requested, please keep yourself muted, because from my side I don't have command to keep you muted. Please keep yourself muted. Out of 100 cases of a particular disease, how many cases are showing this test as positive? This is diagnostic sensitivity. In UK literature, you will find this term as diagnostic sensitivity, and in US literature, you will find this as clinical sensitivity. Meaning of both are same. Diagnostic specificity. Out of 100 cases who are not having a particular disease, how many are showing negative tests? If suppose all 100 are showing negative tests, then the diagnostic specificity is 100%. Sensitivity out of 100 cases of disease, how many are showing positive? Diagnostic specificity out of 100 cases who don't have a disease, how many are showing negative? Positive predictive value from the test side. Out of positive, how many are having this disease? And negative, if a test is negative, how many are not having this disease? Understanding of these terms are very important because in kit literature, very frequently you read all these things. Then when we are buying equipment, desirable characteristics of equipment, what all you expect in equipment, performance specification. Performance specification means what are the claims of company people. If suppose they are saying in this machine, precision for hemoglobin is 2%, linearity for hemoglobin is up to 20 or 25. This is their claim. These are specifications. And whatever you get in your laboratory, when you check precision in your laboratory, when you check linearity in your laboratory, those are performance characteristics in your laboratory. We have to understand this. Linear regression analysis. If suppose you are buying equipment, new equipment, how you can compare that with the machine which you are already using. With this, if suppose you plot the result of a machine, suppose we are comparing hemoglobin. On x-axis, you are writing whatever results you are getting with your old machine, and on y-axis with your new machine. So in ideal situation, you will get a line at 45 degrees. Whatever is coming 5 with old machine is coming 5 with new machine. Whatever is coming 10 with old machine is coming 10 with new machine. But it is very difficult to get ideal situation. But if suppose more or less it is overlapping this line, that means it is more or less providing the same performance. But if suppose like this light line, which is above this ideal line, and it is starting with 5, that means there is intercept. If it is line is parallel to this ideal situation, that means continuously are getting values higher than expected. And the difference is same at all levels. This is called systematic constant error. Now see the line below this ideal. The difference between ideal and this line is increasing with increasing concentration. This is called systematic proportional error. So when we compare, so we are comparing two equipments, so we have to see this. And there is a calculator available on Westgard website. You can submit data of your two machines on that, and that give you the graph like this, and one R2 value. If this R2 value is more than, this is called correlation coefficient. If it is more than 0.9, there is a good correlation. And if it is more than 0.95, this is excellent correlation. It should be as close to 1 as possible. So when we are discussing these terms, we have to understand what are these terms: validation, verification. If you go by the definition of ISO 15189 or any standard document, so it is very difficult to understand what is this validation, what is this verification. And generally they say validation means confirmation through provision of objective evidence that requirement for the specific intended use has been fulfilled. And what is verification? Confirmation through provision of objective evidence that specified requirements have been fulfilled. Now both definitions are looking more or less similar. How to differentiate? In validation, there is some claim. And in verification, there is confirmation of claim. If suppose some company people are saying in this machine precision for hemoglobin is 2% and lower limit of detection is suppose 3 gram per dl and linearity is up to 30 and up to 20 gram per dl. If they are saying so, that means they must have done some studies. So whatever they have done to claim, that is called validation, and those are called validation studies. If suppose they are saying this machine has throughput of 100 tests per hour, so whatever they have done, that is called validation. And if you are checking in your laboratory to confirm their claim, then it is called verification. I don't know how many of you are comfortable in Hindi, but generally I explain this during my presentation in North through this example. So many of you who cannot understand Hindi at all, please excuse me. Otherwise, I have explained. But even if you know little bit Hindi, you will be able to get the message through this example. All of you know the story of Ramayana. Shabri, one character in that story, offered fruit to Ramchandraji. But before offering those fruit to Ramchandraji, she tasted those fruit. What was the purpose? She wanted to be very sure. Whatever I am offering, that is really good for eating. It is not poisonous and it is actually sweet, which should be the characteristics of a good fruit. After tasting, she was sure whatever I am offering, this is good for eating. Lakshmanji, who was real brother of Lord Ram, was sitting next to him. If he would have thought, he is my real brother and this Shabri is outside party, so it is my duty to ensure whatever she is offering, whatever she is claiming, whether this is really good for eating or not. Or you can say, in other words, I have to confirm her claim about this fruit. In this situation, if he would have tasted, what he would have done? In way you can say verification. And what Shabri did, she was claiming, whatever I'm offering you, this is good for eating. Validation. I have explained you validation, verification. You can relate with this example also. Only thing is you have to remember in actual story, Lakshmanji didn't taste that fruit. You have to remember that. Calibration is method of ensuring the desired performance of equipment. There are various ways of calibrating the equipments. For this non-analytical instruments, we take the help of some calibration lab, which is accredited for the calibration of that particular equipment. As per ISO 17025, because that is the document for all testing and calibration laboratories. And your all analytical instruments are calibrated by the manufacturers. Then there are various components of this validation. When you do validation, you have to use quality control. You participate in proficiency testing also. You also see people who will use that method or machine. They have competency. They have ability to use that machine in a required manner. You are maintaining equipment also in a required manner. You are also correlating whatever results you are generating. So you are keeping a relevant record for all these activities as a validation record for verification, because these topics are very big topics and you require at least 1 hour class for individual. But I have included here just for the sensitization. We have to do so many things in these activities. And now suppose somebody else is saying, this machine is good, this method is good. You have to check about accuracy. You have to check about precision. You have to check for the analytical measurement range. And you also have to check about interferences. When you're buying a new equipment, you have to see so many things. First, see whether the space where you will put that machine is adequate or not, and what is the service backup you will get for that machine, extent of technical training required, whether your people will be able to understand all those things or not, or some special training is required for them. Also see instruction manual in terms of clarity and usefulness. If required, ask company people to elaborate on certain things. Linearity means maximum value which you can report without dilution. Also check precision for various analytes. Carryover means a low value sample is affected by previously run high value sample. Check that also. Compare your result with the established method. See performance in aqueous also. Analytical sensitivity means what is the minimum difference that machine or method can detect. Suppose you have 3 machines for glucose. One machine is detecting glucose 110, 120, 130, 140, difference of 10. Another machine is detecting 101, 102, 103, 104, difference of 1. Third machine is detecting 100.1, 100.2, 100.3, 100.4, a difference of 0.1. So this minimum difference which a system can detect is called analytical sensitivity. We have to see what is useful for you. Analytical specificity means it is detecting only that analyte, nothing else. Like in your 3-part differential, in your hematology machine, your nucleated RBCs are also counted with total leukocyte count. Whatever you get, that is not the true total leukocyte count. You have to subtract these nucleated RBCs. This is called analytical specificity. That system is measuring only that analyte. What are the other substances which are also get measured with the analyte which you tend to measure? Analytical time, how much time it will take in one cycle to measure something, and also see throughput time. If suppose after 20 samples, you are able to measure, check 20 samples in 20 minutes, but suppose after that some washing cycle is required, or you have to stop machine for 30 minutes or 40 minutes because of excess heat generated during that analysis, so your throughput time will reduce. You see how many samples you can process in a normal working day. Cost per test, including operating time, reagents, daily startup procedure. All these things add to your cost per test. It is not only the reagent tests. Staff acceptability, how much technical expertise is required. Then you are maintaining instrument in your laboratory. What are the manufacturer recommendation? What all you have to include in daily maintenance, what all you have to include in weekly maintenance, in monthly maintenance, in 3 month or 4 month. Company people, they are coming and they are doing preventive maintenance, AMC or CMC, whatever you want to take. So all these things should be in place. Generally, people say these days, for this equipment records IQ, OQ, and PQ. Sometimes we get confused what is included in IQ, what is included in OQ, what is included in PQ. People who are working in the accredited lab, they don't find all these things in their ISO 15189. When they see different text, hematology people, when they see their Dacey and Lewis, or biochemistry people, they see their Tits and Henry, microbiology people, they see their books, Cruikshank, etc. They don't find these terms IQ, OQ, and PQ. They get confused. So I will tell you briefly about these also. In pre-installation, before installing, IQ means installation qualification, OQ means operational qualification, PQ means performance qualification. Before installation, first, some site assessment, whether the space is suitable for this machine or not. If you require, you can call company people also. You please approve this place for the machine. Then keep copy of purchase order in your file. When you're getting machine, copy of invoice, what all parts you have received, list of that, you check, operation manual. Then if suppose you have to develop some procedure, how you will use that machine in your laboratory, you develop that, and SOPs for various tests which you will perform on that machine. Then if any training package, these company people, they are providing you some manual or some CD or pen drive, keep that also in safe custody. Now, you're installing your machine. On opening the box, visual inspection. You have ordered a new machine. It is actually new. It is not refurbished. It is not damaged. Then see the assembly, what all parts they are when they are joining, how they are joining. You have already checked site and environmental requirement like temperature, cleanliness, not excess heat is there, not excess dust is there, sterility is being maintained. Power check, there is uninterrupted uniform power supply, earthing is proper. Then your company people are providing training to your people. Do the post-training evaluation also and keep all this documentation in place. And also take the details, name and contact number of support service. Till now, you were dealing with the marketing people. Now you have to deal with service engineer. His or her detail. You have to deal with your vendors. Who will provide you reagent? Their contact detail. And diagnostic specialist of company. If suppose you have some technical issue, you want to know something about interpretation of something, then contact detail of the diagnostic expert also. Then this IQ data is approved and reviewed by some authorized person. Then you have installed your machine, but your machine is not ready for operation. Calibration schedule and procedure, maintenance schedule and procedure. We discussed daily maintenance, weekly maintenance, monthly maintenance, how to carry out all those things. Then linearity, precision, carryover, document in your laboratory, then you will get performance characteristics. And you see whether your performance characteristics are close to performance specifications or not. Then check with control. Then check with already tested sample. Now your machine is ready for operation. Regularly run, regularly participate in internal quality control, IQAS. You shall have well-documented procedure. Whenever you are getting any outlier, evaluate, do the root cause analysis, take appropriate action, document it, and monitor for its effective and monitor your action for its effectiveness. Then any maintenance, preventive or repair, shall be documented. Who are the key operators of your machine for a particular period? Their name should be available close to the machine so that you know only trained and authorized persons are operating your machine. Then somebody is approving, okay, we are maintaining all these things, and this is your IQ, OQ, and PQ. Also develop safety protocol in your laboratory. Physical hazard, chemical hazard, biohazard, disposal of biomedical waste as per your biomedical waste guidelines. Sometimes people get confused. When we are regularly participating in IQC, is it essential to participate in IQAS? See, with IQC, we get information about precision, how close our results are to each other. With IQAS, we are getting information about accuracy, how close our result is to true value. Both are important, and these are requirement of ISO 15189 also. If you are going for accreditation, want to comply, this is must. For this internal quality control, how many level you will run, how frequently you will run, this is decided based on your workload and distribution of work. And if accreditation body has decided something, if accreditation body has recommended something, at least you have to follow that. For this IQC, we shall have a documented procedure. We have to implement as per our documented procedure. Then data review. Any outlier, it shall be detected and take appropriate corrective action for the outlier and monitor your corrective action for its effectiveness. So when we say evaluation, we shall have knowledge of these Levy and Jennings chart and Westgard rules. What are these rules and how to decide based on these rules? But in the hematology, situation is little difficult or little different because stability of control is less. Every month, your lot is changing. So you cannot use the data of previous lot for the next month. It is not possible to analyze by LJ chart. So what you can do CV% comparison. In the month of May, for hemoglobin, my CV is 2%. In the month of June, it is 2.1%. In the month of July, it is 1.9%. And suppose in the month of August, it is coming 3.5%. There is significant change. Why my CV has increased so much? What is the reason? You can analyze data with CV% comparison. And for in hematology, if you want to see the variation in the differential count in your 5-part differential, to calculate this CV%, use absolute value rather than percentage, because in that case you will be calculating percentage of percentage. It is better to use absolute value. So whatever CV you will get, that will be more meaningful. In EQUAS, for quantitative, we use Z-score, and for qualitative, we compare with the peer group, whatever these EQUAS provider they recommend based on the evaluation of peer group. We follow that. Important thing is any relevant pre-examination deficiency. What I mean to say, suppose you have collected 1 ml EDTA blood in 2 ml EDTA tube, and the clinician is saying, I'm interested in hemoglobin and platelets also. So you please perform test on this sample. You are performing, but this information shall be communicated to clinician in the report. The sample quantity was inadequate in the vial, although tests were performed on request of Dr. So-and-so. We are taking adequate care of all steps, pre-examination, examination. Now, when we are evaluating our results, we have to take care of certain things like delta check. What were the previous results on that patient? Any interfering substance like lipemic sample interfere with your hemoglobin, TLC. You correlate your result clinically. In hematology, also see histograms. They provide very good information. Also see scattergram, develop understanding of these. Also see numerical data. If suppose hemoglobin is 15 and your hematocrit is 39, there is some problem. There should be ratio of 1:3. So that way, there is ratio between RBC and hemoglobin 1:3 and hemoglobin and hematocrit 1:3. If there is any difference, find out what is the reason. Any suspect flag, all these things shall be seen and interpreted properly. So this is the timeline for COVID-19 testing. So now all of us know how these things are interpreted. From day 1 to day 28, this RT-PCR will come positive. Generally take 5 days to develop symptoms. On 7th day, the IgM will come positive and it will remain positive till 21 days. On day 14, IgG will come positive and it remains for a longer period, and we also don't know how long it will remain positive. In some cases, this level falls shortly, and in some level, it remains high for a longer period. Now, reviewing patient results is important component as we discussed. Then posting patient results is also very important. Because when you're posting results on your website or on your computer, there shall not be any typographic error. Maintain patient records for defined period. Also monitor turnaround time. You're releasing results within committed time frame. And also take feedback from the patient, your customers. They are satisfied with the services. If you're finding any concern, you can take appropriate action on that. And for our laboratory, certain key points. One thing is laboratory should be registered with Pollution Control Board. Then you shall have MOU with your service provider who will collect waste from your lab and also see that service provider is approved by that Pollution Control Board. Follow biomedical waste guidelines as per your national, regional, and local guidelines. And at present, this is very important for any laboratory. Follow latest Ministry of Health and Family Welfare and ICMR guidelines, whatever is applicable to your laboratory. Take good care of, if suppose you're performing this COVID-19 testing, although this applies to all tests, proper sample collection and transport, correct documentation method, proper interpretation of result, take care of turnaround time, whatever information to be conveyed to clinician, inform them, whatever information we send to the regulatory body through their portal or whatever mechanism they have developed, follow that. Now, I would like to express my gratitude towards our professor, Dr. S.K. Sood sir. And we discussed so many things. You may be wondering, who was the person behind all these things? And because right now we have so many different means of communication. We have this internet also, so much information we are getting through internet and other means. This accreditation, it was all started somewhere around 1998, and actual work started, you can say, somewhere around early 90s. Professor S.K. Sood is the man behind accreditation of medical laboratories in India. He thought of all these things, and I am his student. When we were in University College of Medical Sciences, we were doing our post-graduation. There we used to have this documented SOPs in our laboratory. Many SOPs were handwritten, written by Professor S.K. Sood and some other our senior faculty members. And we also used to have one register. In that we used to write clinical history of patient. If we are receiving any bone marrow or any test for any special hematology test, any sample for any special hematology test, is to write history. We were following many things, and when this accreditation was started, we found all those things in this ISO 15189. And he also developed, in fact, he was the chairman of technical committee for the preparation of NABL document 112. In this, NABL has further elaborated many requirements of ISO 15189, and he is also lead assessor of NABL. He started his career from the All India Institute of Medical Sciences. He is postgraduate. His undergraduation was from Gwalior, and he was associate professor of pathology in All India Institute of Medical Sciences. Then he joined University College of Medical Sciences as professor and head. He developed hematology department there. Then he also developed department of hematology in Sir Gangaram Hospital. And when this B.L. Kapoor Hospital was started, so he took the charge of head of laboratory services. He has written a book, Sir has written a book on this ISO 15189. This is assuring quality and competence. I also got the chance to contribute one chapter in this book. This is a very good book, and whatever we have discussed, almost everything I have learned from Professor Sood only. And you will find these things in a simple language in this book also. And if any of you want to purchase a copy of this book, you can take screenshot of this slide. You can take the screenshot of this slide. I will stop here for 10 seconds so that you can easily take screenshot. Then I will again start.
Dr. Jain, we will be sending the information about the book to the all participants.
Okay, right. Actually, what is our aim? As a laboratory person, our aim is to provide a little extra care so that we can contribute to save one more life. Now, what I will request all of you, although we have to go out for various our activities, for our work also, but when some essential is not there, stay at home. Maintain distance whenever you are out, and stay safe. Follow these proper guidelines of hand washing and this physical distancing to keep your body and soul together. Now, thanks to all for your patience, for listening for this last 1 hour. Thank you very much. So now, Mr. Kamal, you can take over.
Thank you, Dr. Jain, for a very enlightening, presentation and the very informative, and I think you're a great teacher. Thank you for wonderful presentation. I will read out to the questions in a minute or so. I also, Dr. Jain, with your permission, Dr. Suneet Sood, who is the co-author of the book, he is also available online. If any person have any specific question related with the book, Dr. Sood will be happy to answer. But I have some questions for you here, Dr. Jain. One is question from Dr. Srijita Nandi, ICMR. What is the frequency of retesting, rechecking as a part of the ECAS where regular PD program or ILC is not available?
See, there is one guidelines by Michael Thompson, International Harmonized Protocol of 2006. They say participation of proficiency testing shall be at least twice in a year. When they say it shall be at least twice in a year, if you are doing this inter-laboratory comparison 3 times in a year, it is more than sufficient. This ILC is also one of the proficiency testing. If formal EQUAS is present, you are participating in EQUAS. If that is not available, then you are doing this inter-lab comparison. So thrice in a year is sufficient. If you want to do more frequently, then nobody is stopping. That's all. Yes, Mr. Kamal.
Yeah, Dr. Jain. Another question is if our hematology controls are old, means open after 1 month, and the results vary. Is any other quality check or check by blood smears? Please advise.
See, if controls are expired, the one thing is we cannot use those controls. That is not acceptable. But suppose if control results are within expiry date, these are not expired, within expiry date, but we are finding variation. And we want to see whether problem is with the control or in our system. So what we can do, in that case, we can test with the retained sample. If suppose we are getting more variation in the morning evening control values, but the sample which has been tested in the morning, and when we are testing that sample again in the evening, there is not significant variation. So that means machine is not showing variation. There is problem in the control material itself.
Thank you, sir. We have time for 2 more questions. Please explain moving averages in Bull's algorithm.
See, this Bull algorithm is not an alternative of internal quality control. This is an additional mechanism of checking variation.
Okay. Last question, sir.
I'm explaining this Bull's algorithm. This is another mechanism of checking variation. What happens? We check for these analytes, hemoglobin, TLC, RBC, platelets for 500 consecutive samples. And whatever average we are getting, suppose for hemoglobin we are getting 12.3, for TLC we are getting 6,400. We are getting 6,400. Then we are checking our daily average. Suppose on August 1st, this average is 12.4. It is okay. On August 2, it is 12.2. It is okay. But suppose one day we are finding 12.9. So, or if it is moving in one direction, on one day it is 12.1, then 12.3, then 12.5, then 12.7. So these are called moving averages. That means something has happened. So we have to check that. This was first described by Dr. Bull. So therefore, this is called Bull's algorithm. Yes.
Thank you. Thank you, sir. Sir, any central Ministry of Health in India is thinking about a central ECA center in India?
Central EQUAS center?
Yeah, in India, any sort of
no.
Is there?
Right now, there is no movement in these lines.
Okay. Last question. So PHC, CHC level labs will be work as under quality control like Gulf countries?
No, I'm not getting this question.
I am, sir. The question is by Mr. Gopal Sharma. We will unmute you. You can ask your question, Mr. Gopal Sharma. Vinayak, please unmute Mr. Gopal Sharma.
Mr. Gopal, you can unmute yourself.
Yes, sir. Just I want to ask, in Central Ministry of Health is going on this direction that they can open a new EQUAS center to all over the country, so they can send the samples to the PSC, CSC level laboratories, because in the government and private sector also, if we are getting like this samples from the Central Ministry of Health or EQUAS center, then we can improve our PSC, CSC level laboratory quality controls. Because I worked in Gulf many years, so we did there like that. Every 3 months we are getting the samples, and we are doing. But here in India, we are purchasing the samples from CMC or like external agencies. We have to buy. Then only we can do. If Central Ministry of Health, if doing in this direction, that is very good for us. Just
But
only my suggestion.
as I answered, but I think there is at present there is no movement in this line. So this is good idea, Dr. Gopal, but
Yeah.
we have to talk to relevant authority in the ministry so that appropriate decision can be taken in this regard.
Thank you, sir. Thank you.
All right. Thank you, sir. Thanks a lot for, as I said earlier, it's a wonderful presentation. You're a great teacher. And thank you all for your participation today in this very, very important and informative webinar by Dr. Jain. We will be sending the information about Dr. S.K. Sood's book shortly to all participants. You can check your email and the registered email ID shortly with that information. Thank you, everyone. Have a great day ahead. Thank you, Dr. Jain.
Thank you, Mr. Kamal.
Thanks a lot, sir. Thank you very much. Thanks a lot.
Okay.